Welcome to Foresight's Biotech and Health Extension Group, sponsored by 100 Plus Capital. Really excited to see all of you here. Maybe I will share my screen for just a moment to bring people up to speed a little bit on where we are in the program. We started this year quite strong. And so what we're doing this year is really always a mix of individual different talks. So we always have a mix of investor Q&As. We already had Karl Pfleger on this, then a policy-focused discussion where we really focus on policy. And now we have our technical keynote seminars again. And as we said already in the introductory meeting, the way that we take notes throughout this year is by in the back creating still the seminar summary. So don't worry, they won't go anywhere, but we're also building out a tech tree that is mapping the space in which you're all operating and a little bit easier to grok level. Okay, so this is just to bring you up to speed on where we are in the current program. Next on up, we have again another investor Q&A with Christian Angermeyer. This is also to say, if you want to join the more back and forth conversation, we have the Foresight Discord here. I won't go on it, but feel free to join this here and feel free to still add yourself as a member here. Check out all the meetings that we've done in the past year. And in the year before that even. So I'll share this spreadsheet again with all of you, but this is just to say we have a backbone here and we have a plan in place. This year we're having lots of individual different meetings. Okay, so this is all on the housekeeping and side of things. For now, I'm incredibly happy to have Vittorio Sebastiano here. We've actually met, well, this is now, I think it was 2019, so quite some time ago when we had an in-person Foresight Biotech and Health Extension investor meeting at IndieBio together with Anastasia and Gialetta. And it was really lovely to have you there. You presented as part of a team of really incredible presentations. I'm sharing a little bit more context on that here in the chat, but we're really, really happy to have you here again. Thank you so much for joining us. Vittorio is from Stanford University, and today he'll be speaking on epigenetic reprogramming of aging, leveraging the principles of reproduction for the treatment of aging-associated diseases. He will speak for about 20 minutes, then I will launch with a few questions. Then we want to hear all of your questions, so feel free to collect them in the chat. And I want to try something new, so let's maybe reserve the last 10 minutes for having a little bit of a back and forth off the record discussion in case you have any updates to share that are only really relevant for people in this group, or in case you want to introduce yourself and you're new to this group, that would be a good way to do that at the end of this meeting. So let's reserve the last 10 minutes for that. But for now, without further ado, I'm really, really excited to have you here. Thank you so much for joining, Vittorio. And I've already had a sneak peek at your slides and they are fantastic. So the stage is yours. I will share more info about you in the chat.
Okay, thank you so much, Alison. Thanks for the kind invitation to you and to the Foresight Group. I'm really flattered and honored to be here today. And I see some familiar faces in the audience, so hi to all those I know. So can you see my screen? Can you see my slides? Great. I just need to move this away. Okay. Yeah, so it's really a pleasure to be here, and I'm happy to kind of share with you some of the work, some of published work that has been done in my lab in the context of aging and longevity. Towards the end of the presentation, I will also maybe if time allows, also speak about a couple of additional projects that are running in the lab. I won't have time to really go deep into the details, but maybe we can also leave it for the Q&A in case. So yeah, generally speaking, kind of the idea of the lab, given my background in developmental biology and in reproductive biology, is how we can leverage the principles of embryonic developments and reproduction and utilize them, kind of leveraging them and kind of biohacking them, and apply them to other non-embryonic, germline cell types. So, well, I think it's pretty obvious for you guys why we are doing this. So I will go briefly over this because I think it's kind of common sense now. Aging, of course, is the main risk for one or more of what you see here, of the indications that you see here. So there's going to be over the next year, a dramatic increase in the number of people that will be at risk just because of the fact that, chronological and biological age increases, there will be an increasing number of people that will be at risk of developing one or more of these indications. If we combine that with the fact that, again, the population worldwide is exponentially growing, and with that also exponentially growing the number of people that will be age 60, 65 or over, this really kind of speaks, and I think that's kind of obvious also from what we're seeing in the, in the VC, in where basically the funding is actually now moving into, this is definitely something that we need to address, and we need to address it fast. So classically, the way medicine has seen this problem has been basically to kind of prevent in somehow or treat symptomatically the diseases that are caused by aging. So if we have this, if we see this curve and we go from birth to death, basically classically the intervention of modern medicine has been to kind of prevent, some of the symptoms, or maybe treat singularly and symptomatically each single disease, right? But I think now it seems pretty clear that now we can take a completely different, dramatically different approach, which is really intervention and making sure that actually these aging per se doesn't happen at the tissue or cellular level. And with that, basically making sure that the diseases that are associated with aging don't develop. And so my lab basically is really focused, has this mission, okay? And again, given the fact that I am a developmental biology and a reproductive biology, by training, I really believe that there is a lot about the biology of development, the biology of embryonic development and germ cells development that can be utilized for the purpose of actually combat aging and extending not lifespan, but health span. So I really want to stress and highlight, this mission. So how? Well, in many different ways, and so I will give you an idea of how we're thinking about this problem. But before going there, I think that I want to kind of state that although there are many hallmarks of aging that have been described, I personally believe that the epigenetic aspect of aging is really kind of the fundamental linchpin of the problem. And I'm not necessarily saying that the epigenetic changes are the cause or the consequence of aging. They could be both, but fundamentally speaking, if you take into account what happens in the cells, cell autonomously, but also non-cell autonomously, fundamentally speaking, all these different phenomena or these different triggers, at the end of the day, affect the epigenome. At the end of the day, affect the way cells behave, what genes they express, what they don't express. And that is, again, fundamentally driven and controlled by the epigenome, which is, this over kind of this program, this information that is kind of sitting on top of the genome, of the DNA information. And we know, of course, that with age, so if we go from young to old cells, that epigenetic information dramatically changes at all levels, okay? We are getting a very good understanding about methylation changes, and we are starting to learn also about other changes, for example, 3D chromatin organization, histone positioning, histone modifications, and so on and so forth. So many new things will happen in the future, but we are starting to get a very good understanding of this process. And of course, there are a lot of questions that kind of come from this concept. Well, the first important one is, well, are these changes stochastic? So can we really think about epigenetic drift? Or is there a program that is, maybe somehow embedded somewhere in some genes that is responsible for these changes? So this is another question that we have in the lab, and Madi and Andrea, two postdocs in my lab, are trying to kind of tackle this question from a methylation standpoint. But that's not going to be the focus of the research of the talk today. But what I really care about is that if it is true, and it is true, that the epigenetic information changes with age, and that aging is fundamentally kind of the linchpin of a number of diseases, if we can tackle or if we can somehow reset the epigenetic information, potentially we could actually really intervene at the root of the aging process. And with that, we could potentially, tackle simultaneously all of these indications. Okay, so big picture, but I think there is definitely reasons to believe that this potentially is possible. So how? Well, let's go back quickly to, and again, I put my developmental biology kind of lenses on, and we know that the epigenetic information is malleable, is tunable, is programmable, right? Why? Well, we know this from the fact that during development, this epigenetic information changes, and it can be completely reset to almost kind of a stage 0 or an age 0 type of epigenetic information. So if you think about, for example, what happens during pre-implantation development, we know that the sperm and the egg come together. Each of those has a specific program, a specific epigenetic information that gets dramatically reset over the course of the first few hours of development. So there is a massive reorganization of the DNA methylation, a massive reorganization of the histone positioning, histone modifications, and so on and so forth. And the same happens also in germ cells. So during germ cell development, there is a similar wave of epigenetic remodeling that resets basically the information of PGCs and germ cells, broadly speaking. Now, this has, of course, has been put in place evolutionary for a very specific reason, right? You want to change one program, namely, the germ cells program, into a new embryonic-like program. So it has a function, evolutionary, and the same is true for germ cells. But what I'm proposing here is that those principles, since this massive demethylation and epigenetic reprogramming is happening, maybe we can, by understanding how that happens, we can actually use it and apply it to the remodeling or the resetting of the epigenetic information that happens with age. Okay, it's not a crazy idea, because actually, if you think about the facts, if you think about, for example, somatic cell nuclear transfer experiments, you're pretty familiar with the Dolly the sheep, Cumulina the mouse, and so on and so forth. That experiment basically shows that if you take a somatic nucleus from any differentiated cell of any age, you take that nucleus and you put it in the cytoplasm of an egg, the egg has all the information to reset that program back to an embryonic-like cell, to the point that now you can make a cloned animal, a fully living organism out of that process. And guess what? The lifespan of that individual, of that clone animal, is for the most part normal. So that means that whatever epigenetic information, meaning age-related epigenetic information, was accumulated in that original somatic cell, that information can be reset completely to 0, to the point now that the new organism has a normal lifespan. And in principle, you can keep doing this, and in some way think about immortality from again from a cellular and epigenetic standpoint. Great. Now, we also know that the same process, in a way simplified, but also, similarly dramatically effective, is the process of reprogramming to IPSCs that was pioneered by Shinya Yamanaka. We know that we can take an old cell, somatic, fully differentiated, and we can make an IPS, so an embryonic-like cell, which is fully reprogrammed and which is youthful by all means. Now, if we take that cell and we differentiated it, for example, in the same original cell type, we get the original cell type, but this cell now is young by all means. Okay. So, and this has been shown, again, in a variety of different ways. So the embryonic cells, or the embryonic-like cells, the IPSCs, and the progeny of those cells are youthful by all means. They have longer telomeres. They have, enhanced mitochondrial functionality. From the epigenetic standpoint, they are younger. They can cope with the DNA damage much better as youthful cells. So the question that we had was, well, this is happening, right? So you can turn an old cell into a young embryonic-like cell, and you can then make that young embryonic-like cells into a young somatic cell. So the question is, what if we skip this process? What if we just try to make an old cell a young cell? Because we know that these two things happen simultaneously. There is a change in cell identity, somatic cell into embryonic-like cell, but there is also an old cell that becomes young. So can we separate these two processes? Can we decouple them? Can we actually work on one aspect of reprogramming without necessarily dealing with loss of cell identity? And long story short, again, inspired by the work of Shinya Yamanaka and the work by John Gurdon, for example, in somatic cell nuclear transfer and IPS reprogramming, we have shown that that is actually possible. And the key is to make sure that you control in time, in duration, and in dosage the window of reprogramming of the cells. All right. So this work has been published a couple of years back. So all the details are there. I will just very briefly go through that. So when we started doing this, I was really kind of focused and detailed about how do we control the reprogramming. Because again, keep in mind that the starting hypothesis was the window of reprogramming, the duration of reprogramming could be the key to decouple the two processes. And so in order to prove that, you would need to be really sure about how long you reprogram the cells and make sure that at some point you really want to shut off the reprogramming process, okay? And so the first question that we had was, well, how do we make sure that we control reprogramming very tightly, very in a very regulated fashion? And so what we decided was to basically we looked at the various reprogramming methods that were out there, and we started to kind of go through them. And we said, okay, what about viral transduction? And we said, well, viral transduction is not really safe. So there is a kind of a safety kind of red flag there, because of course there is insertional mutagenesis if you use lenti and retroviral vectors. On top of that, it's really hard to turn off the genes once they have been delivered into the cells. And the same is true, for example, if you use DOX-inducible vectors, or even if you use AAV vectors, which are a much better kind of solution, but still, they can persist in the cells. So we said, okay, forget about viruses. What about plasmids? So what about DNA-based solutions? Well, safer, but not safe, because there is always the problem of insertional mutagenesis. Every time you insert DNA, double-stranded DNA into cells, there is always a chance that the DNA may insert in an unpredictable way into the genome. Plus, there is always the problem of persistency. So how do you make sure to get rid of the vectors? How do you make sure to shut off the expression of the vectors? So at the end of the day, these two methods are great experimentally, but unfortunately, they really give very poor leverage to the user about the tunability of the system. And we are really leaving a lot to the cells to cope with or to deal with, from transcription to translation, stability, relative ratio of the factors, and so on and so forth. So what we did instead was to take an mRNA-based approach. Why? Well, main reason, because it's safe. There is no way that an mRNA can insert into the genome. There is no likeness of expression, meaning that, as long as you provide the mRNAs, the genes are expressed. But if you stop providing them, the genes are no longer translated, and so that the factors are no longer expressed, unless, of course, there is an endogenous activation of genes that could be responsible for progressing, through the reprogramming process. It's also a very fast approach, very efficient, and most importantly, it's very tunable. And by that, I mean that basically you can simultaneously and orthogonally work on a number of different, parameters, and really try to come up with the most optimal cocktail and dosage, for controlling the reprogramming. So you can control the number of factors that you're dealing with, the types of factors, their sequence, their stability and cytotoxicity, the relative ratios of the factors, how long the mRNAs can survive or can live in the cells, the amount, the biodistribution, and most importantly, my research is always kind of clinically focused and translational in a way. Most importantly, and we're learning, actually this was a gamble, but it was a good gamble. We're learning this now, through the times that we're living, through COVID. There has been an explosion, in terms of how you can implement mRNA for gene therapy. And so the mRNAs are easy to formulate, they're easy to be made, and probably in the next few years we're going to see gigantic factories of mRNAs that are going to scale this process up and make it affordable and cheap. And also from a GMP standpoint, it's a much simpler kind of approach to use. So and so at the end of the day, basically what's left to the cells, once you have figured this out, what's left to the cells is just to translate those optimal mRNAs and let them do their job, right? In addition to that, we also had a very long experience in mRNA-based IPS reprogramming. So we really knew a lot of details about the whole process of reprogramming. And we figured out that actually, the first few days of 2 to 4 days of reprogramming are those days where loss of cell identity hasn't happened yet. And that was the window where we basically focus our studies to figure out if any change in age was happening in the cells. So long story short, and I will go quickly through the details, but long story short, we have shown that using a cocktail of mRNAs that we can transfect into the cells, we can turn old, exhausted cell into youthful, functional cells that are young across pretty much all the hallmarks of aging, with the exception of telomeres. We can probably talk about this, in the Q&A section. Most importantly, we have shown this in human cells, and I think I can still claim that we are really truly the only ones so far that have published work in human cells. And this protocol has worked across many different naturally aged cells. That's another important distinction that I want to make, because sometimes when you use genetic models of aging, those are great, but they only partially recapitulate the complexity from a physiological standpoint, transcriptional standpoint, and epigenetic standpoint, the complexity of aging, as a phenomenon. All right. All right. So, well, we just to walk you very quickly. So we have done this analysis, a very comprehensive, a very holistic analysis, because we think, since aging is such a complex phenomenon, we wanted to make sure, and since we think that the epigenetic is the fundamental linchpin that controls a number of different hallmarks, we wanted to make sure that this process was really working, comprehensively, across many hallmarks. So we took a variety of, we used a variety of different assays, for example, from epigenetic information, physiology, mitochondrial function, and so on and so forth. So just to very quickly walk you through the data. So we took whatever possible single cell quantitative measurements across thousands of different cells to get population-wide information, but also single cell information. So we took that information and we combined it, in a kind of, broad general graph. And you see here that, for example, for H3K9 trimethylation, you see that with age, the global level of that mark goes down dramatically in fibroblasts on the left side and in endothelial cells on the right side. The treatment, which consists in only 4 days of reprogramming with the optimized cocktail, changes that profile globally in a youthful fashion, in a youthful fashion. So going in the direction of youthful cells. And that's what you see here in light blue or cyan, both in fibroblasts and in endothelial cells. Now, with that in mind, we looked at a variety of different hallmarks. And you can see for yourself that every mark, every hallmark, or every kind of feature that changes with age gets reestablished in a youthful fashion after the treatments. Epigenetic marks, autophagosome formation, protosomal activity, mitochondrial membrane potential, mitochondrial ROS, sirtuins, and so on and so forth. Now, is that due to the fact that you're stressing the cells? No, because if you deliver the GFP mRNA in the same amounts, those hallmarks or those assays do not change, or if they change in the other direction. So they make the cells even, older in a way. Then we looked, of course, holistically at the transcriptome profile, and we saw that with the treatment we reestablished a youthful-like, gene expression profile in old cells. We look at the methylation clock, of course, in collaboration with Steve, the guru of the methylation clocks, and we also saw that even at the methylation level, we can reestablish a more youthful methylome. And then we said, okay, that's great, but we're working with somatic cells. From the clinical standpoint, this doesn't really make a lot of sense, right? You don't want to make younger fibroblasts and then maybe reinject them into the skin, right? But if the same approach holds true in stem cells, which are the cells that are responsible for the long-term regeneration of a tissue, then maybe we could really have a very long-lasting effect due to the transient reprogramming. And so in collaboration with Marco, who is a friend and a colleague, and Tom Rando, we basically tried to apply the principle, to stem cells. And of course, when it comes to aging, the problem is always, what model do you use? That's kind of a bottleneck in the field. So what model of aging do you use to prove what you want to prove? So fortunately enough, Marco in Rando's lab had developed previously a way to isolate stem cells from fibers and keep them quiescent. So, retaining their full stemness in vitro for about 48 hours. So that was the perfect kind of, model for us, because we could isolate the cells, keep them in culture, maintain their stemness, and put them back in vivo as soon as possible after a short treatment. So that's what we did. We started with mouse cells. So we took the fibers, we isolated the stem cells, and we divided them into two groups: the untreated group and the treated group, from old and young mice. The treated group received the treatment for just 48 hours, so just 2 pulses, and then the cells were tagged with luciferase and GFP and transplanted into, in this case, a young host immunocompromised mouse. At the time of transplantation, the muscle, the receiving muscle, was also injected with barium chloride, which is a cardiotoxin that basically destroys the muscle. And so basically what you're doing is you're challenging the transplanted cells to regenerate the whole muscle, okay? So that was the experimental layout. And in some groups, in some mice, we followed them for about 11 days. And in some mice, we did a second injury 2 months post-transplantation to, again, measure if that kind of treatment had a long-lasting effect on the regenerative capacity of the stem cells. The untreated cells were transplanted on the left leg. The treated cells were transplanted on the right leg to make sure that they were in the same systemic environment, because that could also have an effect, an impact on the ability of the cells to regenerate. And here you see the data. So that over the course of 11 days, this is how the transplanted, untreated, aged stem cells repopulate or rebuild the muscle. So that's the bioluminescent signal that you see. You compare it with young, untreated stem cells. Of course, the young, untreated stem cells are much more, much more efficient in doing so, in growing. But, the aged, treated cells behave like, if not better, the young stem cells. We did morphometric analysis to make sure that actually this growth was not due to, kind of a crazy growth or tumor-like growth, within the cells. Remember that our claim is that the cell identity is maintained after the treatment. And what it was very exciting to see that actually we couldn't see any tumor. We could only see fibers, muscle fibers. There were more fibers and there were thicker fibers, suggesting 2 things. Well, first of all, that the treatment seems to be safe. So there is no loss of cell identity. There is no, all of a sudden, cells that are going crazy and forming tumors in the cells. So safe approach. The second is retention of cell identity, because the muscle cells could only generate, what they're supposed to generate, which is fibers at the end of the day. And here you see the quantification of that. So this is the number of fibers here, and this is the average thickness of the fibers. When we did the second treatment, 2 months after, of course, the bioluminescence goes down because, of course, now you are destroying the muscle. Okay, there's only stem cells pretty much left there, and it was really mind-boggling to see that the old treated cells behaved again like young untreated cells, suggesting that there was memory. So this cell autonomous treatment was leading to a very long-lasting effect that, despite the environment around the cells, could still kind of elicit a growth that was like young stem cells. In this case, we did an experiment with aged mice because, of course, the environment could make a difference. And in this specific case, we went all the way through 30 days, and we measured in vitro the contractility, so the strength of the muscle. And you can see here very dramatic, changes. So that's the force of a young untreated muscle. That's the force of an aged untreated muscle. If you take an aged muscle, you transplant it with aged stem cells, you restore the physiological function of that muscle, which is aged. But if you take an aged muscle and you transplant it with aged cells that have been treated, you restore the physiological strength of a young untreated muscle, which is 40% roughly force induction, which is pretty dramatic. We repeated the same experiments with human cells. So we took muscle biopsies from patients. Same experiment. Of course, this was done on a shorter timeline because of paucity, of course, of biological material, but same result. So the cells grow like young untreated cells. And what's really interesting is that when you start staggering the samples by age groups, you see that the most dramatic difference that you see is when you start dealing with with samples coming from older individuals, suggesting again that there is maybe epigenetic changes happening even in the stem cells that can be somehow reset with the treatments. So, kind of to finish the first part, basically, if we think about development, as a bidimensional kind of, set of coordinates, on the one hand, you have the function, on the other hand, where you have the age. You start from embryonic, which is very young, but also non-functional, meaning that it doesn't have a function as of yet. And you go progressively through an increase of function and an increase of age, right, with time. The classical interventions can only bring the ball back or the marble back just a tiny bit because you're just tackling one small aspect of aging. But what we think we can do with epigenetic reprogramming of aging is that we can, bring back in time, but not in function, or even in function, actually, sorry, in time and in function, we can bring a dysfunctional old cell to a functional younger cell. And importantly, the same cell type. So, of course, this approach is scalable, in principle, for the same reason why you can make IPSCs from any cell type. I think that you can make younger cell type from any cell type, starting cell type, as long as you know how you control that duration, how you control that reprogramming window, again, in time and in duration. So there is a number of studies in my lab that are ongoing, with many collaborations about these different cell types and these different indications. So I don't know if I think I'm already kind of at 30 minutes.
A little over time, yeah.
Yeah, I don't. Maybe I can leave this second part, for the Q&A, and maybe share with you some other ideas that we have in the in this context. And so let me just go to the acknowledgement then, which is of course the people in my lab, the funding, my institutes. In particular, I want to thank Jay, who was the spearheader of the whole epigenetic reprogramming of aging project, and now in particular Andrea and Madi, which are working on kind of follow-up studies on that group. And thank you for your time, for your attention. I'm happy to take your questions.
Wow, fantastic. Okay, well, I just from you showing the slide for a second, I'm hoping that we get to that in the Q&A. I'm sure that people will be very interested. But let me launch into the conversation maybe with 3 general questions, and then we leave it up for the Q&A. And so a few things that we want to ask every person who's presenting here is just to really get a clear outline for people that are maybe even new to the field. And three questions. First one is, what is an exciting goal of your field? Which enabling technologies would you like to draw on to make progress? And the third one, what is an unsolved challenge? So maybe let's start with the first one. In a nutshell, what is a really exciting goal for your field? What would you be excited about if we achieved it?
Well, I alluded to that already, during the talk. I think that most exciting thing is I think we're really at a very nice inflection point where we can really make the difference for people that are actually have a health span that is severely affected, people that live in conditions for a very long time in conditions of frailty. And I think we are really, really at the inflection point to make a difference for those people. So I'm excited to think about the prospects that this technology can deliver to those people and to me in about 20 years from now. So I am also very, very egoistic, in that regard.
Yeah, the earlier the better. Then the second one is, which enabling technologies from other fields maybe even would be required to unlock new capabilities that you think are along the line?
Yeah, so in my opinion, I think that the RNA technology, broadly speaking, is gonna be really a game changer for what we're trying to do, but also for other fields. Just see what happened with the vaccines. It was really dramatic what the mRNA technology could do in the context of vaccines, and the same will be true for gene therapy, and of course for reprogramming, together with the delivery kind of so liquid nanoparticles, so the in vivo delivery solutions for mRNAs. So those two things, I think they go hand in hand. Those are going to be really the game changers in the future.
And could you once more specify maybe which specific capabilities would then stand to be unlocked next if you had those on hand?
Sorry, what do you mean by that?
If you had those on hand, let's say, we're making massive progress in those, what are specific things that could be unlocked next? What is on the horizon, just inside?
Oh yeah, gene therapy, for example, correction of genetic disorders, just by providing, for example, to specific cell types, the gene that is missing, to perform this cystic fibrosis, for example, just to name a few. Even for cancer therapy, gene therapy, broadly speaking, for all the genetic disorders. And then, of course, this new area, which is epigenetic reprogramming of aging. Yeah.
Lovely. Okay. And then finally, what is the challenge that you would like to see solved? If you now had significant capital to allocate, and you're like, I really just want this particular thing to be solved.
In vivo delivery. So I want in vivo delivery to be solved, because if we figure out how to effectively deliver to specific cell types what we want to, the gene of interest, this is going to be a dramatic shift in the way we think about regenerative medicine as a whole.
Lovely. Okay, that's all for me. Now we get 2 questions from the audience, which there are a ton of. So first one up, we have Johnny. Do you want to go? Johnny Adams. We can also take Carl first if you're still getting ready. Carl, are you already in the pipeline?
Yeah, I can go. Great to rehear the talk. It's great work. I love it. So I want to ask a broader, simpler question about reprogramming in general. Right, there's sort of widespread acknowledgement that the recipe is a little tricky. Right, you have to get the timing exactly right. All that I've heard so far is that the recipes are just do this, fixed recipe, do this for this amount of time at this dosage. When are we going to move towards a more sophisticated recipe that has conditionals based on readouts from the cells being reprogrammed themselves, right? Like do this until you see this change, and then dose for 2 more days or whatever, or then change the dose to X or whatever, right? Because we have imaging techniques that we can do on cells that are non-destructive, and we now have there's new work sort of coming along on like longitudinally sampling in non-destructive ways too. So where do you think that's going? Are people working on that?
Yeah. There's plenty of people working on that. We are working on that question as well. It's a great question, because there's 2 ways of looking at that problem. On the one hand, you can say, okay, is there a universal cocktail that, for example, you could use for different cell types, and the only thing that you need to figure out is the timing? I think that's the case for the quote-unquote Yamanaka factors. I mean, we're using an optimized version of the Yamanaka factors. It's not just the 4 canonical. So again, the idea there is very simple. The same cocktail can be used to reprogram any starting cell type to IPS. So that means that you can make any starting cell type a younger cell type. And again, by controlling the duration. And this can
But the timing might be different for different cell types.
The timing could be different for different cell types, absolutely. But it's a cocktail that could work across cell types. The other way, the beauty of this is that you can use it also as a discovery platform. And so maybe there is also fundamentally a universal logic about rejuvenation, which is triggered by the Yamanaka factors, but then, kind of leads to another set of factors that could be cell-specific, or maybe they are not cell-specific, that you could learn and utilize at some point in combination or as a replacement to the Yamanaka factors. So, but this potentially could be cell-specific. So at that point, you have the counter problem, which is like, okay, if I want to rejuvenate chondrocytes, I need to use this cocktail. If I want to rejuvenate hepatocytes, I need to use this cocktail. Or maybe not. Maybe we have another version of universal factors that can work across cell types that maybe also from a safety standpoint could also be in a way less alarming, because of course people always argue about MYC and other,
Okay, so let me ask a more specific question then. Like,
sure.
Are people in your lab looking at the cells while the cocktail is being applied and trying to identify changes that predict whether or not it's going to work, and whether, when you should have to disable the cocktail, things like that?
Yes. The problem though there, yes, absolutely. We're using what the technology now allows us to do, of course. There is something that I think is missing. To go back to Alison's question, actually, there is another point I want to make. We need to develop tools to measure aging in a quantitative fashion, and hopefully also in a non-invasive fashion. So I think that AI here could play a very, very fundamental role, and we're trying to work on that as well. So we're using molecular biology tools right now because those are the ones that we have, but we want to basically feed that information into a process that could actually tell us if and how the cells is going through reprogramming, when to stop it, and then learn from that process. Yeah.
Okay, Carl's happy. Lovely. Next one up, we have Anton. Are you ready?
Yeah, I'm ready. Actually, my question is already partially connected with your previous answer. So in case where we know the exact cell type, for instance, like in your case in muscle regeneration, so there were papers on applying differentiation methods to also facilitate wound healing in muscle. So my question, do you think that the differentiation approaches, when we don't go dangerous Yamanaka, but just go one step, like from neuron to neural progenitor, did you actually compare Yamanaka-like reprogramming with the differentiations? And do you know what are the limits of the differentiations that you cannot solve without actually adding Yamanaka?
Another great point. So I think that this idea of rejuvenation can be used in two ways. One way is to keep the identity fixed, of course, by knowing how long you reprogram the cells, keeping the identity fixed and just rejuvenating the cells, so making it more youthful from a physiological standpoint. Another way of thinking about this is that if you push the boundary a little bit more, but again, not long enough to bring them back to IPS in vivo, but long enough to bring them back to, a kind of a slightly earlier stage in development, for example, to a stem-like, cell in vivo, that can also have, of course, the rejuvenation side aspect of it, but it can also have the regenerative potential, associated with that. So, for example, David Sinclair and work on the regeneration of the optic nerve, or the recent work by Thomas Brown on the regeneration of the heart, using the transient reprogramming. Those are two beautiful examples of, if you know for how long you can push the cells back in time, you can reestablish a function that is not necessarily lost with age. Well, it could be accelerated with age, but can also be lost just by the virtue of the developmental trajectory of the cells. So in other words, to make it simple, the heart cells don't regenerate. They lose that ability to regenerate the heart, right? The same is true for neurons. At some point in life, postnatally, the neurons can no longer regenerate. They can no longer grow. But they had that ability when they were fetal-like. So if you can understand that process and bring them back in time, to the point that they were fetal-like, but still neurons, or maybe neural progenitor cells, or maybe neural stem cells, then you can reestablish that regenerative capacity as well. And this could have really dramatic impacts on the regeneration of the tissues, because you wouldn't have to rely on IPSCs, which are great, but come with the cumbersome of redifferentiating the cells back into the final cell type that you want. And those protocols are often very tedious, very complicated, and very costly. Did I answer your question?
Yeah, so you didn't compare, as I understand, because your approach, as I understand, you have like Yamanaka-like mRNA, so modified Yamanaka. And if like we compare, for example, we know which transcription factor can get from neuron to stem-like neuroprogenitor. So it is not your approach, it is different approach. But in both cases, we increase regeneration potential, but it's actually not clear in terms of age. Do we know that age change is less when we go just one step behind, not like your way where you just...
I see what you mean. No, yeah, the same idea could be used. For example, if you know what makes a neural stem cell, what really defines, a neural stem cells in principle, and some works are already there. You could potentially provide that specific cocktail to a neuron, and you can turn potentially a neuron into a neural stem cells. Period. So that is possible, absolutely. Now, is that changing the age of the cell? That's still something that needs to be figured out. I can tell you, for example, that if you take other types of cocktails, for example, cocktails that can turn a fibroblast into a neuron without going through the IPS, right? It's called transdifferentiation. So you horizontally convert a fibroblast into a neuron. There is at least a couple of examples there showing that you can convert the function. So again, you turn a fibroblast into a neuron, but from an aging standpoint, you haven't done nothing. So the age of the cell remains the same. So the Yamanaka factors, broadly speaking, I think they have also this kind of resetting of the aging, information, which cannot be achieved or may not be achieved with other cocktails, which are more cell specific in a way. Does it make sense?
Yeah, thank you very much.
Lovely. Okay, next one up we have Sonia.
Thanks, Allison. Great talk. So I'm curious about the telomeres. They don't seem to be affected by your technique at all. What does this say about the relationship with aging? These things matter or not?
So I have two answers to your question. So in my opinion, my humble opinion, I don't think that telomere attrition is such a big deal for human aging. It's a big deal for senescent cells, but senescent cells explain only a fraction of the aging phenotypes that we see at the systemic level. So that's the first answer to your question. So I don't think that telomere attrition again or elongation is such an important. But again, the mRNA technology is so plastic that if one day we will discover that telomeres are a big deal, you can add the TERT to the cocktail, and you can also again by virtue of transiently expressing TERTs. And I think there are already examples of that by Helen Blau, Helen Blau's work, for example, that you can just transiently express TERT, you elongate the telomeres, done. So that's the first answer. The second answer is, I think that for what we're claiming here, it's actually very, very good to see that we don't see telomere elongation, because telomere elongation is something that happens late during the reprogramming, when the cells have already converted to a stem cell-like kind of, a phenotype. So that is a sign for me of loss of cell identity. And so it's good that we don't see telomere elongation. But again, if we want, we can add TERT to the cocktail.
Great.
Lovely. We have Mia next. Let's see if we probably still have time for this question, and then we do intros at the end. Mia.
Well, Vittorio, thanks. That's a beautiful presentation, beautiful progress. And I agreed with your comments about the telomeres wholeheartedly. So I have two questions. One is not very serious, but your titles are, you're in childhood and mothers and in pediatrics. Could you have your own establishment that doesn't have that, but there's maybe back to pediatrics, back to childhood, right? You're looking like a pediatrician. But my serious question. So you showed that with the mRNA, with the variety of mRNA that you chose, you trend to get younger, but you didn't achieve really what stem cells do, where they erase aging. So is that because you didn't use the all of the factors, or did some cells really erased aging? Why is it that there's a trend and not a total annihilation of aging?
Great question, Nir. Thank you. So I think that's so keep in mind again that we don't want to change identity. So you can apply this idea to different cell types. You can apply it to fully differentiated, committed cells. And what you want to do there is just to make them younger without changing their identity. So again, pushing that cell back to a stem cell-like could be a way of thinking about this, but you have to extend the reprogramming process. And you may get in that, it's called area 51, which is, when the cells don't really know what they are, because there's still a ton of Yamanaka factors, around. They are receiving signals from outside and they don't really understand what they are. And so they may actually go into another trajectory all of a sudden.
Yeah.
So keep that in mind. But the same is true. So if you apply the same idea, not to fully differentiated cell, but to stem cell. So if you know how to isolate them, for example, put them in culture, treat them, and of course QC them, you can actually, as we did for the muscle stem cells, if you target a specific population of stem cells, you can get the same rejuvenation, and at the same time, you also kind of take advantage of their regenerative potential. Now, think about this in vivo. If we could figure out a way to specifically target the stem cells in vivo, for example, through very fancy liquid nanoparticles or epitopes or aptamers or antibodies on the surface of the LNPs, and we can specifically target the stem cells, at that point, you're hitting two birds with one stone. You're making the stem cell younger, you are doing it specifically in the stem cells, and by doing so, you can have a very kind of long-lasting effect, because you are targeting the population of cells which is responsible for the long-term regeneration of the tissue.
Thank you.
I see. Great. I see a nod there. We have Matthew. If you're very quick, then maybe we can take your question too before we move to intros.
Yeah, it's fast. As Vittorio knows, I'm a huge fan of in vivo delivery and all these things. But my question is actually really simple. If you just rejuvenated cells from, say, a muscle population and put them into an otherwise unharmed individual, would you see a benefit? Is it likely to have therapeutic benefit to do this on its own, or does it really require the injury to see a difference?
Great question, Matthew. Thank you. It's we don't know that yet. Again, for now, we have challenged the cells, or we have challenged the system to kind of be an aging-like model or an aging-like. So I don't know if you just treat the cells and you put them in a normal environment, what would happen to the cells. It's a good point. I don't know the answer to that question. It's something that we should probably look into. But on the flip side, though, at some point, you can even think about this you can think about this in two ways. One way could be restorative, so you fix a problem. On the other hand, you could think about this as a kind of preventative, approach. So you prevent aging from happening in the sense that you make sure that the cells do not, even though they're not aged yet, they don't progress, they don't age at the same pace as if they weren't, they were untreated. That doesn't make sense what I'm saying. So what I'm trying to say is that you can even think about at some point as pulsing, these factors or maybe other factors, pulsing them in a way that now the cells are not going through the aging process, and maybe they are more resistant to the aging, to the triggers that would lead them to age. Does it make sense?
Yeah, I can think a skin market would be pretty attracted by this idea of pulsing them back.
Yeah.
But thank you, Vittorio.
Sure.
I'm glad we arrived there. One final bit before we move into intros. What could this group do to help you work along?
Well,
this is an open discussion about what could we do? If people get excited about this, what can they do to help you?
I think that well, for sure, kind of the network is very important, because I think feedback from people working, for example, in other fields is extremely important. For example, AI, machine learning, ideas on how to implement, that's I think always valuable. And the other, as an academic, the usual answer is like funding, in the sense that these are very expensive and experiments that require substantial funding. And so in that sense, that is also very helpful for me and for the community. I think I can speak for every academic in the group.
Well, I was going to let this be just a chat question, but since you kind of touched on it, do you want to expound on any of your opinions on how the recent influx of money and big multi-billion-dollar efforts in this specific area are going to change the epigenetic reprogramming sub part of the field?
Well, I mean, it's pretty obvious that it is. I mean, you have read the news, right? There is a tremendous kind of influx of capital in specifically on this, and I'm in a way very, very flattered that and happy that we were one of the first one actually to pioneer this whole idea. So it's rewarding in a way. There is hundreds of millions of dollars now that are going into this, and there's still a ton of work to be done, from the academic standpoint. So I think that, of course, biotech is great, because it can accelerate dramatically the speed of the final product. But at the same time, I think that a lot of money should also still go into research, basic research, to understand the mechanism. So yeah, I think that what's happening in particular here in the Bay Area is really a signal that is definitely something worth investing into and going after.
Lovely. That's a great note to end it on. Thank you so much for joining, Vittorio. That was really, really lovely. Good to see about just how much progress there has been making and what rapid pace. I would also love to perhaps welcome 3 new members in this group. One of them is actually in your lab. So perhaps we start with Madi. Madi, if you want to introduce yourself briefly, and they were fine with being on the record.
Sure. Yeah. Thank you, Alison. My name is Madi. I'm the computational biologist at the Vittorio lab. We had collaboration with Steve Horvath. We developed an AI-based method to measure epigenetic changes in aging and in any rejuvenation treatment. And I like how Steve calls it the 4th generation of aging clocks. So this tool helps us to understand the mechanism of rejuvenation at a molecular level. And it also helps us to optimize the rejuvenation treatment like timing and factors, as Carl mentioned about. We already applied it to dietary restriction and rapamycin and reprogramming. We are looking into T-cell exhaustion and epigenetic changes. So if you have any rejuvenation treatment that you're working on and want to measure or understand epigenetic changes with your treatment, please reach out. We might be able to look into it.
Lovely. And I shared the Discord link here in the group chat for people to just contact each other. Next one up, we have Alexei. Alexei, welcome.
Hi, thanks a lot for organizing the whole group. I think it's very important for us to move things forward. So a couple of words about me. I'm more of on the business side of things. I was an early team member of In Silico Medicine. I was an early team member of Centaura, that's related to aging. Now I'm a co-founder and CEO of a company called Grey Matter. We are developing therapeutic peptides for longevity indications. We're based basically on an in silico technology, which allows us to make very safe therapeutic peptides, and we believe safety is paramount for longevity. That's why I thought that it would be a good idea to make a longevity company based on that platform. We also have a fish facility. So if you are interested in, and we have notobranchius species, which are very short-lived fish with 4 to 6 months lifespans. So if you're interested in conducting trials, we can organize something with regards to notobranchius. And I also should mention why I joined your group. I think last couple of months, I started to think very deeply what sort of companies should exist for longevity, but do not yet exist. And I asked several people of where I can find people who also think about it. And basically, Jean Herbert, he advised me to join you, so join I did, and very nice to see familiar faces and new faces. So I'm very, very happy to be a part of Foresight.
Welcome. It's really lovely to have you on board. And last but not least, we have Andre.
Thank you, Vittorio, for the talk. I just transitioned from industry to academia. I worked in Giro for a pretty long time. It's an AI data-driven biotech startup in longevity. And now I work in Vadim Gladishev's lab as a postdoc. So I'm a physicist. So I'm trying to make sense of aging as a physicist and try to kind of distinguish what's actually kind of deterministic in aging, what's stochastic, and what's like better approaches to rejuvenate and to make sense of the data.
Very cool. Wow. Okay. Now you have a few, I guess, faces to the names. And so I'm hoping that encourages all of you to reach out to each other if you have more questions or want to collaborate. Thank you so much for staying 5 minutes over. I'm German. I hate being late on time, but I think it was worth it. Vittorio, we have to have you back at one point to discuss the second part of your presentation because it looked too good to be missed. So sure. I hope you don't mind reaching out again. Okay, everyone. It was lovely to see all of you. Next one up is an investor Q&A with Christian Andermeyer. And so thanks all for joining. And it was really lovely to see all of you.